The idea behind the study is really not just the idea of linking a gene to a disease, but it's taking that linkage or that ability to say this gene is lost in the disease and then translating into a clinical outcome or effect. What we were able to do in our preliminary work was to show that this MTAP gene is dysregulated in a large percentage of the sporadic osteosarcoma samples that we examined. It was a small number, but in a majority of these samples the gene was dysregulated. Specifically, the splicing of the MTAP gene, the ability of the messenger RNA, how a gene becomes a protein to the functional end in a cell, how it's actually stitched together is somehow altered in these tumors. That in and of itself is an interesting finding, but what becomes more interesting and what becomes important to the population of individuals with sarcoma or the doctors treating them is that this gene MTAP is an important gene in how a cell takes certain products and converts them into other products. There are drugs now available and in experimental phase that can target this gene. We believe, and what we'd like to show, that these drugs may have more or less effectiveness depending on how this gene is spliced together. So the ultimate goal of our studies is to identify these tumor samples, look at this gene, and then characterize specifically what kind of a tumor the individual has. Do they have an osteosarcoma or a bone sarcoma with or without this MTAP gene dysregulation? Once we know that, can we use specific drugs to target these two types of tumors? So I guess really the important finding here and the important goal or outcome is for us to be able to say not that an individual just has a sarcoma, but what type of a molecularly defined sarcoma do they have? Because we believe this may have therapeutic implications.